In this tutorial, we're going to learn how to grow an uncontaminated culture of bacteria in the lab. Bacteria are found almost everywhere on Earth. There's bacteria on our hands, on surfaces, and even in the air. For that reason, scientists need to know more about these organisms in order to prevent the spread of pathogens and cure diseases they cause. Scientists need to culture these microorganisms in order to study them. They grow them in large numbers so they can be seen as colonies. To culture microorganisms, we must provide them with nutrients, including carbohydrates as an energy source, various minerals, a nitrogen source so they can make proteins, and sometimes other chemicals. In addition, they need warmth and oxygen. It's worth mentioning that you need uncontaminated culture of microorganisms to investigate the effect of chemicals such as disinfectants and antibiotics. It's important to work in a sterile environment to avoid contamination coming from our surroundings, as well as you need to sterilize equipment sterilized in a special oven called an autoclave. It heats the equipment at a high temperature and pressure. Keep in mind that you must take extreme care when culturing microorganisms, because even though the bacteria you'll be growing is harmless, there's a risk that a mutation, which is a change in DNA, might occur and produce a new dangerous pathogen. Now let's see how to prepare an uncontaminated culture of microorganisms in the lab. But what are the equipment needed? Well, for this practice, we need a culture medium. It can be either a broth solution or agar gel containing nutrients. Agar gel is heated and poured into a petri dish, then left until it solidifies. However, the nutrient broth solution is a liquid medium. We also need an inoculating loop, Bunsen burner, tape, marker pen, and a bacterial broth culture containing the bacteria we want to grow. As for the steps, first of all, sterilize the inoculating loop by passing it at an angle through the flame of the Bunsen burner until the entire length of the wire turns orange-red. This kills all the contaminants on the wire. Then, allow the loop to cool down for a few seconds to avoid killing the inoculums. Now to remove the inoculum from the broth culture, hold the culture tube in one hand and in the other hand hold the sterilized inoculating loop like a pen. Remove the cap of the culture tube with the little finger of the hand holding the loop. Do not put the cap down or else it becomes contaminated. Flame the lip of the culture tube briefly to force air out of the tube and prevent airborne contaminants from entering the tube. While keeping the culture tube at an angle, insert the inoculating loop and remove it. Again, flame the lip of the culture tube and replace the cap. Now to transfer the inoculums into a petri dish, lift the edge of the lid just enough to insert the loop. Streak the loop across the surface of the agar gel in a zigzag pattern. Be careful not to scratch or break the agar while streaking the loop. Keep it parallel and do not apply pressure. Remove the loop and close the lid, and finally re-sterilize the loop. Remember to secure the lid with adhesive tape so that nothing goes in and out, but only oxygen can still come in. Do not tape all the way across the Petri dish. This will allow harmful anaerobic bacteria to grow. And label the Petri dish with the initials of the microorganism you are growing, your initials, and the date of incubation so that you do not incubate it for a long time. Once you inoculated your plate, place it upside down in a heater container called an incubator for 48 hours at a maximum temperature of 25 degrees Celsius. It is placed upside down to reduce the possibility of condensed water on the lid to drop on the agar, causing fluid to run on the agar medium. College and school labs incubate the culture at 25 degrees, which is lower than the body temperature, to reduce the risk of microbes growth which are pathogenic to humans.